Respected Rajkumar sir, other teachers and my dear friends, a very good morning. I welcome you all to my presentation on the topic Safety Indicator Organisms, Pathogenic Ecoli. These are the various topics we are going to learn in this presentation, like introduction, history, type of indicator organisms, and what do these indicator organisms indicate, the use of E. coli as indicator organism, scientific classification of E. coli, virulence properties of E. coli, isolation and identification, mechanism of intestinal pathogenic E. coli strains, and the challenges of using E. coli and current trends of E. coli as indicator organism. So here is the introduction. That is the indicator organism that actually reflect microbial quality. Organisms or their metabolic product whose presence in given food at certain level may be used to access existing quality or to predict the shelf life of the product. So indicator organisms are bacteria that are used as a sign of quality or hygienic status in a food, water or environment. So historically these conditions have been related to in sanitation and public health concern. Over the years, however, the use of indicator organism has been extended to provide evaluations of quality in addition to safety of the particular commodities. So here's talking about the history. Indicator organism term was first coined by mid to late 1800s due to various waterborne diseases caused by the consumption of poor quality water. Isolation of pure microbiological culture, that is Salmonella typhi, the causative agent of typhoid fever, which was discovered by Carl Ebert in 1880. Theodore Escherichia isolated Bacterium coli, later was known as Escherichia coli, in 1885 from a fecal source and stated that it was native resident of the human gut. Coliform test was regarded as the most appropriate means of assuring the sanitary status of drinking water supplies in 1914 by the US Public Health Service. So worldwide, the acceptance as safety and hygienic indicators was increasing. So here is the type of indicators. There are mainly two types of indicators, that is safety indicators and quality indicators. Safety indicators means that suggests that a microbial hazard may exist and use is intended to minimize the risk of exposure to the hazard. So mainly E. coli is considered as a safety indicators, which is suggested by the International Commission on Microbiological Specifications for Food, and thus it is applied to water safety. And E. coli possesses the desirable attributes of an appropriate safety indicator, and its presence suggests the fecal contamination and potential entry of pathogens into the system. Whereas in quality indicators that are used to assess issues important to product acceptability, example shelf life, organoleptic characteristics, spoilage, etc. A good example is the yeast and mold determination, which can serve as a quality indicator for the cereal grains. Indicator organism as a single microorganism or group of microorganisms may be pathogenic or non-pathogenic in nature. Its presence reveals a potential problem with quality, hygiene and safety of the food product. It shows the microbiological status of water and food, as their presence indicates the possible presence of ecological similar pathogens. So what does indicator organism actually indicate? It mainly signifies the potential presence of pathogens. A labs in sanitation has required in good manufacturing practices, that is GMPs or a process failure. It mainly influences the consumer acceptability of a product. It also indicates the conditions of storage and many other factors like intrinsic, extrinsic, chemical, physical factors that might influence the microbial growth, process history and storage conditions of the product. And this picture shows the indicator organisms, which is the total coliforms, which is an gram-negative facultative anaerobic bacteria, which is an indicator of the fecal pollution, and the fecal or thermotolerant coliforms, which is known as the pathogenic E. coli, which mainly indicate the fecal contamination. And the other indicator organisms are fecal streptococci, bifidobacterium, clostridium perfringens, sulfite reducing clostridia, etc. Use of Escherichia coli as indicator organism. So these Escherichia coli are the predominant member of the facultative anaerobic portion of the human colonic normal flora. This is mainly present in large intestine of warm-blooded animals, that is endotherms, and since E. coli with some exceptions generally does not survive well outside of the intestinal tract, it's present in environmental samples, food or water usually indicates a recent fecal contamination or poor sanitation practices in food processing facilities. The population of E. coli in these samples is influenced by the extent of fecal pollution, lack of hygienic practices and storage conditions. The mere presence of E. coli in food or water does not indicate directly that pathogenic microorganisms are in the sample, but it does indicate that there is a heightened risk of the presence of other fecal-borne bacteria and viruses, many of which such as Salmonella species, hepatitis A virus, etc. For this reason, E. coli is widely used as an indicator organism to identify food and water sample that may contain unacceptable levels of fecal contamination. E. coli is considered a more specific indicator of fecal contamination than fecal coliforms, since the more general test for fecal coliforms also detect thermotolerant non-fecal coliform bacteria. The E. coli test recommended by the United States Environmental Protection Agency, that is EPA, confirms presumptive fecal coliforms by testing for the lack of an enzyme which is selective for the E. coli organism. This test separates E. coli from non-fecal thermotolerant coliforms. Here comes the scientific classification of Escherichia coli. The kingdom is Bacterium, phylum Proteobacteria, class Gamma Proteobacteria, order is Enterobacteriales, and the family belongs to Enterobacteriaceae. The genus is Escherichia, species Escherichia coli. The Escherichia coli is commonly abbreviated as E. coli. It is pronounced and named after its discoverer. It is a gram-negative rod-shaped bacterium which is commonly found in the lower intestine of warm-blooded organisms. So mostly those E. coli strains are harmless, but there are some pathogenic E. coli strains such as serotype 0157H7 that can cause serious food poisonings in humans and are occasionally responsible for the product recalls. The harmless strains are part of the normal flora of the gut and can benefit their host by producing vitamin K2 or by preventing the establishment of pathogenic bacteria within the intestine. In fact, various classification schemes for coliforms have been emerged. The earliest were those of the McConkie, who recognized 128 different coliform types, while Berge and Dehan identified 256. By the early 1920s, differentiation of coliforms had come to a series of correlation that suggested indole production, gelatin liquefaction, sucrose fermentation, and Voges-Prosker reaction were among the more important tests for determining the fecal contamination. These developments culminated in the IMVIC, that is indole, methyl red, Voges-Prosker and citrate test for the differentiation of the so-called fecal coliforms, soil coliforms and intermediates. Water sanitary engineers, however, require simple and rapid methods for the detection of fecal indicator bacteria. Hence, they are similar to identify coliform group despite being less fecal specific and broader, for which this Escherichia, Klebsiella, Enterobacter and Citrobacter was considered the most among the common genera. These all genera was targeted. So here it shows that there are harmful E. coli strains and also harmless E. coli strains. These harmful E. coli strains are called the virulent strains, which can cause the diarrhea to the healthy adults as well as to the children. More virulent strains which are common in E. coli is the 0157H7, which causes serious illness or death to the elderly. So E. coli is gram-negative, facultative anaerobic and non-sporulating. These cells are typically root-shaped and are about 2 micrometer long and 0.5 micrometer in diameter, with a cell volume of 0.6 to 0.7 micrometer cube. It can live on a wide variety of substrates. So here in this picture you can see that there are total coliforms, fecal coliforms, E. coli and pathogenic E. coli, which are very harmful. Next is the virulence properties of E. coli. Enteric E. coli are classified on the basis of serological characteristics and virulence properties, which is shown in the table. Here comes the classification of E. coli. The first one is enterotoxigenic E. coli, which is a causative agent of diarrhea without fever in humans, pigs, sheep, goat, cattle, dog and horse. And these strains produces two types of proteinaceous, that is heat liable toxics and heat stable toxic, which is LT and ST. And these type of strains are non-invasive, which means they cannot enter the host cell. And these are the leading bacterial causes for diarrhea in children in these developing countries. And the second type of E. coli is the enteropathogenic E. coli, which is the causative agent of diarrhea in human, rabbit, cat, dogs and horse. So this is invasive, which means they can enter the host cell. The third one is entero-invasive E. coli, which is found only in the humans, and this causes a syndrome that is identical to shigellosis with profuse diarrhea and high fever. And the fourth one is enterohemorrhagic E. coli, which is found in humans, cattle and goats. And the most famous member of this virotype is the strain O157H7, which causes this diarrhea and no fever, and it also causes sudden kidney failure, and it is moderately invasive. And the last one is enteroaggregative E. coli, which is found only in humans, and this also causes this watery diarrhea without fever. And this type of E. coli is non-invasive, which produces hemolysin and an heat-stable enterotoxin similar to that of the enterotoxicologic E. coli. Here comes the mechanism of intestinal pathogenic E. coli strains. They are divided into two, that is non-invasive and invasive. In non-invasive, they are again divided into toxogenic. In toxogenic, it comes under the enterotoxigenic E. coli, which is again divided into heat liable and heat stable. In invasive, they are again divided into non-toxogenic and intoxogenic. So in non-toxogenic, there are two types of E. coli, that is enteropathogenic E. coli and enteroinvasive E. coli. In toxogenic, there is only one type, that is enterohemorrhagic E. coli, and then comes Shigella-like toxin, that is verotoxin. So here is the isolation and identification of E. coli. There are different methods for the pathogenic strains of E. coli and non-pathogenic strains of E. coli. So in pathogenic strains, some of these pathogenic strains will not produce acid and gas from lactose. It has also been shown that growth in medium containing the sodium lauryl sulfate and growth at 44.5 degrees Celsius can cause a loss of plasmid, known to encode many virulence factors associated with the pathogenic E. coli strains. So one of the studies shows that 95% of this E. coli cell lost the plasmids during the selective enrichment culture. Therefore, the methods commonly used for the detection of E. coli as an indicator organism should not be used to attempt isolation of pathogenic strains from the food or water. So the methods are different. Isolation of enterohemorrhagic E. coli O157H7 must be approached differently than using the methods for isolating other E. coli strains. And the E. coli O157H7 has some biochemical differences from most of other E. coli strains that can be exploited in isolation and identification methods. E. coli O17H7 ferments sorbitol slowly or not at all and does not produce functional beta-glucuronidase, whereas most of the other E. coli strains are positive in both the tests. So further, these E. coli strains do not ferment rhamnose on agar plates, whereas 60% of the non-sorbitol fermenting E. coli belonging to other serogroups ferment rhamnose on agar plate. So several methods such as DNA probes and polymerase chain reactions, ELISA procedure utilizing the monoclonal antibody specific for an outer membrane protein of E. coli O157H7 and media that can be tested both sorbitol fermentation and beta-glucuronidase activity such as sorbitol McConkie agar containing the mug can be used for isolation of this organism. The identification and enumeration of E. coli of sanitary significance lays upon isolate conformance to the coliform and fecal coliform group definition. E. coli isolates are traditionally identified by their IMVIC pattern, that is plus plus and minus minus in type 1 and minus plus minus minus in type 2. So in this scheme, I refer to the ability of the organism to produce indole from metabolism of tryptophan, and M, which indicate the ability of the organism to ferment glucose to a high acid, as detected by the methyl red pH indicator dye in the medium. VI stands for the production of the neutral products 2,3-butanediol and or the acetone from the glucose metabolism, which is known as the Voges-Prosker reaction, and the C, which represent the ability of the bacterium to use the citrase as is all of the carbon source. So recent data indicate the defining of E. coli by the IMVIC profile is inadequate for the identification of E. coli strains which do not give IMVIC reactions corresponding to either biotype 1 or biotype 2. The relatively high incidence of the type 2 E. coli in some specimen is partly explained by the fact that many isolates require 48 hours to produce a detectable amount of the indole. Hence, additional tests were essential for the specification. So here, this picture shows the positive and negative reactions in each test for the identification of E. coli. This is the biochemical properties of E. coli. The different types of E. coli shows the different result in each of the tests. So here are some challenges of using E. coli as an indicator organism. That is, as soon as the coliform test came to widespread acceptance, complications with its use and interpretation began to emerge. One concern was the discovery that a variety of microorganisms that read positive in the coliform test were not of the fecal origin, and also they are needed at the higher incubation temperature, though the disease-causing strains of E. coli species, which is isolated from the tap water, drinking water, and many other sources, the examination of pathogenic E. coli was not so easy. And there is also no biochemical marker that can separate pathogenic from the non-pathogenic strains, and that found very difficult. To the degree that the naturally occurring microbial pathogens became a significant public health concern, completely new test procedures have to be developed. So these were the challenges. Next is the current trends of E. coli as indicator organism. While the fecal coliform test has its limitation and problems, it also has many attributes. One of that is it acts as a regulatory tool. It has worked long and well. In case of the water quality regulation, coliform testing has been successfully used. And also this fecal coliform test will continue to be the basis for much of the regulatory decision making regarding both the quality water harvesting and the contact recreation. The primary basis of using cultural tests in isolating E. coli as an indicator organism has been overcome by using the PCR, which detects both live and dead bacteria. The PCR is a rapid and reliable tool for the molecular-based diagnosis of a variety of infectious diseases. PCR analysis for screening drinking water and environmental samples has been reported and has been utilized to identify the E. coli in primary water specimens, stool specimens, and outbreaks. Hope you all understand this presentation on this topic. Thank you.